Let me welcome everyone. This is a very good morning to everyone in Americas. Good afternoon for people in Europe and Africa, and good evening for people in Asia and Oceania. Welcome to this third Albrecht Müller ARRIGE Seminar. A few words about Albrecht Müller to begin with. He was one of the few members, such as some of us, that we gathered in 2017 and 2018 with the idea of launching this association that was meant to promote the responsible use of genome editing techniques. He unfortunately passed too early. He was passing over the next year after we founded this association. He was a member of the ethics committee in Germany, and because of that, we decided to devote this series of seminars in his memory. And so this is the third edition of such seminars, and this one, it will be devoted to human therapeutic applications of gene editing. I thank all panelists, all speakers that have accepted to attend this seminar, and in particular, I thank my colleagues Francois Hirsch, Jennifer Merchant, and Julian Kinderer, because they will be helping me running this webinar. Francois will be responsible for the first roundtable, and Jennifer Merchant will be responsible for the second. So we're living, I guess we all know, we're living very exciting times. It's 10 years since we started using these genome editing tools. January 2013 were the first demonstration that these CRISPR tools could be used in a variety of applications. But today we know that there are more than 200 patients, perhaps 300, by the way, in which they have been already receiving these genome editing tools in a variety of formats. Being the first generation, this is the classical CRISPR-Cas9 tools, or the second generation, which are the base editors, or even the third generation that soon will be jumping into the clinic, which are the prime editors, the quality editors. We have a number of different diseases that have already been explored, commanded by the sickle-cell anemia, which probably will be the first treatment that will be approved after all the different clinical phases have been proceeded with success. But other than sickle-cell anemia or sickle-cell disease, we have a number of cancer immunotherapies in which CRISPR is part of the therapy to inactivate some of the negative regulators of the immune system, and also cystic fibrosis, and also hypercholesterolemia, and other genetic basis disease. So we are not thinking that this will be happening in the future. This is happening now. So we're living this present, and this is the time now to reflect, and this is why we thought it was appropriate, it was adequate from ARRIGE to launch this Albrecht Müller Seminar devoted to this topic. Now we will have to evaluate, meaning the society, meaning the regulatory agencies, the European Medicament Agency or the FDA, or the similar agencies in other countries. That's the time when they will have to evaluate in more close details the safety issues regarding the precision and regarding the efficacy of this attempt. So ideally, we want only the gene that has been corrupted or that is not functioning appropriately to be modified, to be returned to a sequence that is mostly carried by most of us in most of the human population. But at the same time, we don't want other parts of the genome to be altered. And we don't want the gene that we want to modify to be modified in different ways as we plan. So these are the issues that we have on the table. And these are the issues that have been dealt with in the different clinical phases. We have cases and we have examples in both sides of the Atlantic Ocean, in Europe and in the US. And I guess we have here delegates from both the US and Europe, from the academia and also from the industry, and also from more a research perspective or from a more ethics perspective. So we will be looking forward to hear what you have to comment on all these different issues. I don't want to take more time. I think it's in the interest of time. I think it is better if we can start. So I will pass over the word to my colleague, Francois Hirsch, and he will be presenting the members and the panelists for the first roundtable. Francois, whenever you want. Thank you all.
Okay. Thank you, Luis. So do you hear me properly?
Yes, yes.
Okay. Okay, so hello to everyone. It's a great privilege to share this session with 3 speakers who agreed to take on their time to present the results on the therapeutics advanced with genome editing. Of course, given the limited time of a webinar, we made a choice. As Luis said, many diseases may now be treated with GE, but as you will see, that I think we did the right choice. Allow me just a personal consideration before handing over the floor to our speakers. In 2015, our interim CEO, Professor Yves Lévy, who was both fascinated by GE technologies, but also worried about their quick expansion, asked Hervé Schneeweiss, the interim ethics committee president, which by the way is the ARRIGE vice president, and can't be present today. We start at the in some ethics committee. We start thinking on the ethical dimensions of genome editing. In a correspondence that we published in Nature in 2017, we called for launching a monitoring group of relevant stakeholders to promote open debate on the societal aspects of these technologies. And in the meantime, we went to Argentina, Latin America, Africa, Brazzaville, and India, New Delhi, to discuss all these issues with our local colleagues. This would eventually lead to the creation of ARRIGE. During the ARRIGE kickoff meeting in Paris in March 2018, some of you were present. I do remember the vibrant messages from 2 mothers of children with genetic disorders, who urged the scientific community to develop G-based therapeutics as fast as possible. And here we are today, in about a decade, to present some of these approaches. So let's now start the session with André Shulika, and thanks for joining again. André is one of the founders of Selectis, one of the, I think, the first company developing genome editing approaches. And he served as chief executive officer in the company inception in 1999. Dr. Shulika worked as a postdoctoral fellow in the Division of Molecular Medicine at Boston Children's Hospital, where he was one of the inventors of nuclease-based genome editing technologies and a pioneer in his activity in the analysis and use of meganucleases to modify complex genomes. After receiving his PhDs in France, in Paris University, he completed a research fellowship in the Harvard Medical School Department of Genetics. And lately, in June 2019, Dr. Shulika was appointed to the board of directors of the Pasteur Institute. André, you have 20 minutes to present what you do in Selectis, and thanks again for joining.
Well, thank you very much, François.
Yes, you can.
Sharing the screen. Let me know if you're seeing the screen.
No, exactly. Now you can present, André. Go ahead.
Okay, wonderful. Okay, well, thank you very much, and thank you for inviting me. Luis, I'm so sorry. I'm not going to talk about CRISPR. Is that okay, or I can jump?
Fine. We are dealing with genome editing tools.
Okay, it's fine, but it's not CRISPR. Anyway, you have to read this slide. So a quick overview of the company. We're, as described by Francois, we've been founded almost 24 years ago. It will be December 1999. So like in 1 month from now, it will be 24 years. We have 3 clinical trials. We've dosed currently more than 50 patients. In total, we dosed, I think, between us and our partners, over 200 patients in total with gene-edited therapies, which is, I guess, probably more than 300 what was cited. One of the things I'd like to point out is that we manufacture everything internally. I think it's something that is extremely important into this industry, especially the cell and gene therapy. Manufacturing is a huge differentiator, and we have a series of partners. So what we do is we have like an end-to-end platform. So we have our own gene editing platform. So we do TALEN, which is the nucleases, but we will do also tau-based editors. It's both either nuclease or base editors. We do both. And I'm going to describe a bit of the platform. One of the main progress that we have essentially in allogeneic CAR-T is targeting hem-onc. We have 7 products that are currently in the clinic between us and partners. And there are solid tumors. There are 3 preclinical assets. There's 2 solid tumor trials with our partners. And we have a series of other things. I'm going to say a few words about our hematopoietic stem cell gene surgery platform for sickle-cell disease and lysosomal storage disease and other in vivo gene editing for liver disorders. So allogeneic CAR-T. Why people do allogeneic CAR-T and why gene editing is so much important? First of all, autologous CAR-T has been a revolution in oncology. And now like I have a series of products that have been commercialized between Abecma, Yescarta, Kymriah, Carvykti, etc. However, what we see is that there is a limit, which is apheresis center. Apheresis center becomes a real gouging problem and limiting market access problems. A lot of patients cannot be dosed. And for example, Carvykti that gives you over 80–90% of complete response for multiple myeloma. I think 3 or 4% of the patient that could be eligible to Carvykti can be treated because of this problem of gouging apheresis center. For example, in the United States, there's 150 transfusion centers that can do apheresis, and well, they cannot treat all the patients. So like there's a real line of patients waiting for this. Plus, at the top of this, currently there's autoimmune diseases using CAR-T that are coming, and it's all autologous. So allogeneic or off-the-shelf CAR-T makes the sense, first of all, because it's scalable manufacturing. We can do hundreds of doses with one batch, if not thousands. There is a robustness, so the consistency in each patient, same product that is given, and the reproducibility and the robustness of what every patient gets is something that is extremely important. And finally, market access. There's also control in production and the cost for patient and also safety and profitability. So that's the platform that we have currently, and that maybe I can try and find a pointer, laser pointer here. Oops. What we do, so what the gene editing part is on this left side, we do a track knockout to remove or minimize risk of GvHD. You will see that there is no GvHD in most of the patients that we dose. And the second thing that we do is that we knock out all those CD52 with TALEN to give a resistance to a preconditioned regimen with alemtuzumab. And on the other side, so that's what on the left side is what we remove from the cell. And on the right side is what we introduce in the cell, which is either dual CAR-T CD20 or CD22 or CD123 for different type of diseases, ranging from B-cell malignancy, B-cell LL, or AML. So how it works, we start with the frozen leukopak. Then we transduce these cells that we saw. We transduce the cell with a lentiviral vector that gets in there, that brings the CAR inside the cell. Then we do a gene editing using a TALEN, which is coded with messenger RNA. Or if we want to make gene insertion, we either bring single-strand DNA or AAV inside the cell to induce, for example, targeted recombination or targeted replacement in genes. And then finally, we expand the cell for a certain number of days. And then finally, there is a purification. The purification is essentially focused on removing every TCR expressing cell to remove totally the GVHD. And finally, there is a fill, finish, and freeze phase. And these vials are stored and can be stored for many years, over 6 or 7 years of stability so far. And then they can be shipped to cancer centers and be provided to patients. So a bit of an overview of the studies we have. So at start, you always have to go through a dose escalation. So we have currently a design which was either using a preconditioning regimen with fludarabine and cyclophosphamide, or fludarabine and cyclophosphamide and alemtuzumab. Remember, alemtuzumab there is a resistance for cell for alemtuzumab by the removal of CD52 by gene knockout using gene editing with TALEN. And then we go from different doses, so DL2, DL2I, DL3, and then we have a confirmation at the time for the dose that we give. So we've given DL3 with a production that is P1, and then now we're dosing with the product that has been manufactured internally, which is P2, with fludarabine cyclophosphamide, and we are going to provide some data at the American Society of Hematology meeting this December at ASH 2023, this December, and probably next year we'll provide some data on the dose level 2I. So when you look at the characteristics of the patient that have been dosed, they range between 17 to 61 years old. They all have BLL recurrent with genetic abnormalities, 42% of them. There is a CRFL2 rearrangement in 21% of them. And all of them had prior lines of therapy that failed, either blinatumomab, inotuzumab, CD19 CAR T for close to 50% of them, or HSC transplant, which is also 50% of them. When you look at the safety, there is no DLT that had been found up to DL3. ICANS, no ICANS, no treatment emergent adverse effects, so 0. Essentially, 11 of the patients had a mild to moderate cytokine release syndrome, which is between grade 1 and grade 2, which is a very good sign because you know that the cells are expanding. There is 0 grade 3 or higher CRS, and 1 patient experienced grade 2 GVHD, skin only, and that was not confirmed by biopsy, and was essentially in the context of reactivation of prior allogenic bone marrow transplant from the prior donor transplant that was given to this patient. When you look at the results, essentially, you see that there is expansion in these cells under FCA, and you see that 50% of all of our overall responses at DL3, 3 patients out of 6 went into complete remission or morphological leukemia-free state, which is MRD negative. So you see, when we reach DL3 with the regimen that is the P1, so the process 1 that was manufactured by our CMO, we have 50% ORR, which makes it already in this refractory or relapsed population, a product that could be already approved. Few words about the new product that we have that is extremely interesting, UCAR-T 20x22, which is the dual CAR-T. The inclusion area criteria are 18 to 80 years old, essentially mature B-cell non-Hodgkin lymphoma, except CLL or SLL, enriched SLL and Burkitt lymphoma, the tumor has to be positive for CD22 and or CD20. So, and a lot of these patients have received prior lines, including CD19 CAR T if eligible. And the primary objective that we have in this study is safety and tolerability, which is MTD, or finding the RP2D, which is the phase 2 recommended dose for UCAR-T 20 by 22 protocol. There is also a DL1, so with those patients at DL1, then we're increasing to DL2 and DL3. DL1, it's a flat dose at 50 million cells per patient. And then we increase to 150 million cells per patient for DL2 and 450 million cells per patient at the end. And that determines the MTD and the dose expansion for large B-cell lymphoma at the end. The preconditioning regimen is fludarabine, cyclophosphamide, and alemtuzumab at 60 milligrams total over 3 days that is given to the patient. So what we see as of July the 28th, 2023, we have 3 patients that were treated at dose level 1 and were available response. Patient 1, 76-year-old female with double expression for diffuse large B-cell lymphoma, relapsed after refractory at CHOP, and radiation therapy for polatuzumab and vedolizumab. She achieved a partial metabolic response at day 28. Second patient, 65 years old female with triple hit transformed follicular lymphoma. She was also received series of prior dosage and did not respond to bendamustine and rituximab, and achieved complete response at day 28. Third patient is an 18-year-old female, so like younger one than the 2 others, with relapsed or refractory transformed marginal zone lymphoma with previous failed chemotherapy, venetoclax, ibrutinib, and bendamustine plus rituximab, and you see that we have a complete metabolic response at day 28. This is very interesting because when you give to the cell to the patient 50 million cells as a flat dose for patients that are over 50 kilos, with a preconditioning regimen that is alemtuzumab, you know that there will be a depletion approximately of 10 million cells by alemtuzumab. So that's a very low dose. You already have 100% response rate in this, which is very encouraging. One of the things that you would see is that the cell would go, and this is the number of cells that you have per microliter in patient at day 14, for the second patient at day 11, and day 12 for patient for third patient. So you see that there is a strong like expansion of cells, and you see that essentially you CD8s that expand, a bit of CD4s, essentially a CD8 expansion that correlates also to the ferritin expression, and it's also correlated to the decrease in number of malignant cells in the patient when we inject these cells. The thing that is interesting is that the white blood cell count come back to normal. All the cells are the allogeneic CAR T that have been injected to the patient, and that makes it extremely spectacular for the current stage. So as a conclusion, you have like the UCAR-T platform takeaway from like 220 patients. There's a track knockout result that is safe, non-reactive UCAR-T, so there is no GvHD by the knockout of the track. The expansion works well for the CD52 with a preconditioning with alemtuzumab in lymphodepletion that has the potential to provide a safety and efficacy and controllable therapeutic window. When we see the comparison between either with or without alemtuzumab, we don't see any negative or any type of increase in infection or risk given to the alemtuzumab preconditioning treatment. There is encouraging clinical data in LL. I didn't show the data for ML given the time that we have, and not Hodgkin lymphoma. There is safety, so the profile on par with approved autologous CAR T therapy on the safety side. And then finally, we have a very strong efficacy, anti-tumor activity consistent with autologous products, but with robustness, with 100% success. For example, for DL1, for UCAR-T 20x22, which is the first allogeneic CAR T in this space. A few words about the future in this space. So we're going to increase the complexity. So there is mesothelin CAR T, where there are 3 knockouts, which is TCR, always CD52, plus TGF-beta R2, to prevent a loop, a negative loop of TGF-beta1 that can inhibit the cell in solid tumor context. There's UCAR-T FAP1 that would have a TCR or MHC knockout also, in order to make the cells stealth to other T cells. And then finally, the most sophisticated CAR, which is UCAR-T MUC1, that is a smart CAR where we replace beta-2 microglobulin by HLA-E, will knock out TCR alpha, will replace PD-1 by IL-12 and knock out TGF-beta2R. So there are 4 knockouts and 2 knock-ins plus the add of the MUC1 CAR, which make it one of the most sophisticated CAR T that will essentially be developed in series of solid tumors, including, for example, triple-negative breast cancer or ovarian cancer. Now, a few words about the gene editing platform. So we use, excuse me, but we don't use like CRISPR for multiple reasons. Actually, we like TALEN for the sequence recognition driven by protein-DNA interaction, which helps us to redesign the protein forever in order to have a very high specificity and activity on site where we want to do. There is a high specificity of targeting provided by the 2 amino acids within each module, so we can precisely at the base pair almost target the CAR T, which is not the case of other gene editing technology. It doesn't give it the precision of TALEN. And finally, the release of DNA ends gives accessible DNA repair mechanism with the same speed on both sides, which allows very much a DNA repair that is very balanced on both sides of the DNA, because the 2 TALEN wrap up with the same KD on both sides of the DNA and allow, for example, the exonuclease is 5' to 3' to unzip the DNA and allow the gene targeting that is more accurate. So currently we're developing also an HSC genome surgery platform where we essentially edit HSPCs. So we take the HSPCs, an autologous therapy, we edit them and then re-inject them in the patient. So there is a high modification efficiency, high viability, primitive HSC, and we can have clinical scale manufacturing that's like this. So we're going to work actually on lysosomal storage disease, MSP1 gene therapy, where we take cells that have IDUA deficiencies, we take cells that are mobilized, we do TALEN gene editing, and then we re-inject autologous bone marrow transplanted cells after have fixed safely and efficiently by gene editing IDUA and let the cells expand. One of the things that we will do, so it's like essentially the gene of interest that will be inserted in myeloid locus at HSPC cells. So the myeloid gene differentiation allows us to make a tissue-specific expression in myeloid cells. So you see that, for example, HSPC does not express the gene of interest, while differentiated myeloid cells will express that. So this technology called ARRIGE allows us to make a very cell lineage-specific expression of the gene of interest. We can also have, for example, MSP1 expressed in proof of concept in vitro, in vivo. So you see the mock and the edit cells for HSPC at CD45 plus cells. And when injected in the brain, like the cells edited in the brain of the cells, so you see that essentially HSCs that are CD45 plus express as well the transgene, the CD11B, and microglia express it, but the CD11B that are negative does not express the cell. So that gives us a very tissue-specific edited efficiency and graft and differentiate in the brain and expresses the gene of interest in the village lineage in a very specific manner. So a quick conclusion, because we're getting at the end. So Selectis have a core technology with the mRNA-based TALEN gene editing, high activity and precision efficiency. It enables cell and gene therapy, various modality for CAR T platform, HSC, genetic disorder, but also in vivo gene editing. We can leverage major players such as we have Servier, Allogen, Ivance, Cytovia, Primera. Recently, we announced an agreement with AstraZeneca. And then finally, we have clinical validated TALEN. Between us and our partners, the real number is 270 patients that have been administered with product derived from Selectis TALEN technology under 10 different investigational new drugs. And one of the most important points is the full GMP manufacturing is now all done in-house and optimized and processed in timeline for plasmid, RV, AV, and IP, and all the other final IMPs. With that, there is 17 or 16 minutes left. We're based in Paris. We have labs and research. All our research lab and platform is also based in New York, on First Avenue and 29th Street. And then finally, we have a manufacturing plant in Raleigh, North Carolina. With that, I would like to thank you all for your attention.
Thank you so much, Andre, for keeping the time and for these impressive results that you presented, and showing that it's not only CRISPR that can be used. I think it was important to show that to the colleagues that there is a life beside CRISPR in a way. So I propose to keep the question at the end of the three presentation. And so if you agree, we move to Denise Dalcara. So it's a colleague from INSERM. She's a research director and a team leader on gene therapies and animal models of neurodegenerative diseases at the Paris Vision Institute. To finish this session, it's my pleasure to introduce Michael Santos, who already kindly contributed in the first Albrecht Müller meeting on Gene Drive. Remember, quite a long time ago now. So Michael is Senior Vice President and Chief Population Health Sciences for the Foundation for the National Institute of Health, FNIH, where he leads Gene Convene Global Collaborative for the better development of genetic biocontrol technologies. Before joining the FNIH, Michael led some programs on malaria, tuberculosis, and HIV at the Bill and Melinda Gates Foundation, and oversaw projects on global health, energy, and environment in both the public and private sector at the Boston Consulting Group. Michael, the floor is yours for 20 minutes. Thanks.
Great. Thank you so much, Francois. Can you hear me okay?
Yeah, good.
Excellent. Well, great. Thank you. I really appreciate the opportunity to talk today about genome editing for sickle cell disease, and my gratitude to the organizers for providing this opportunity. I think this, I hope this talk certainly will dovetail nicely with what Denise just presented. So just a quick outline. As was mentioned in the introduction, sickle cell disease certainly is one of the really promising human genome editing applications. And so I'll give a little bit of an overview to the disease also because I think it's important to some of the challenges that are part of the development of products in this area. Then talk a little bit about the approaches for genome editing for treatment for functional cure or potentially durable cure. And then challenges and opportunities, like I said, for access to these products and further applications. So that I know many people are familiar with sickle cell disease. Just to give a quick overview, of course, it's recognized by the characteristic sickling of the red blood cells. The primary symptoms for people with sickle cell disease are anemia, sickle cell crises, increased infection risk, but there are many more symptoms. The presentation of sickle cell disease is highly heterogeneous, both across individuals and for a given patient across their life course. There are many serious additional conditions such as stroke, increased risk during pregnancy. And so it's a relatively complex presentation. It's not totally simple, but relatively simple genetic presentation. There are variations, but the most common sickle cell disease is caused by this point mutation. And individuals who are homozygous for this mutation have sickle cell disease. If you're only carrying one copy of the mutation, then you have what's called sickle cell trait. Sickle cell trait generally does not have any clinical manifestations, except that it does appear to confer some protection against malaria, which we'll come back to in a second. Sickle cell disease has been, has been known for a long time, over 100 years, and the genetic basis has been understood for over 70 years. It is a disease that has received relatively little investment in terms of research and in terms of access to care to existing methods compared to the burden. Also, I won't be talking about other hemoglobinopathies, but in particular, of course, of note, there is a beta thalassemia product that is FDA approved in the US that is similar to some of what we'll be talking about for sickle cell. So in terms of the epidemiology, here's the geographic distribution of the sickle cell alleles measured in indigenous populations, so not people that have moved from their ancestral locations. And so, of course, you can see this high concentration of high prevalence of the alleles in sub-Saharan Africa and a little bit in West and Southern Asia. If you, from that same paper, and I'll just flash to it, overlay the geographic distribution of malaria before malaria interventions became widely available. Of course, particularly in sub-Saharan Africa, you can see the pretty strong overlap between this distribution of malaria and the presence of the sickle cell, high prevalence of the sickle cell alleles. And that, again, is believed to be related to a selection effect for sickle cell trait in areas where people were under high pressure from malaria. Consequently, sickle cell disease occurs mostly in people of African descent, also some in South Asian and Latin American populations. And this is part of the under-resourcing that I described previously. These populations, certainly in the US where I live, have received disproportionately little attention, both from provision of healthcare and also for research into conditions that disproportionately affect them. Of note, in some of these populations in West Africa, the prevalence of sickle cell trait can be 40% or more. And so here, just in terms of incidence and prevalence, again, because when we think about scaling these interventions, I think these numbers are important. So from the global burden of disease work, the latest incidence estimate is about 500,000 births with sickle cell disease every year. And globally, almost 8 million cases and annually about 34,000 deaths. So of course, for a genetic disease, a monogenic disease, it's quite high prevalence. So the way that it's diagnosed, there are both blood tests and genetic tests, of course. Risk-based prenatal screening is something that's available commonly in high resource settings. So risk based on ancestry primarily. Sickle cell disease is also part of routine newborn screening in many high resource settings, though I think it's really important to know that early diagnosis is not generally available in the highest prevalence settings and lower resource settings. In terms of currently available therapies, so the one curative therapy that is available is hematopoietic stem cell transplant for patients for whom there is a sufficiently good match. Of course, there are a lot of challenges to that procedure, and so only a small fraction of patients are a good match, literally or more generally, for that kind of treatment, for that kind of therapy. There are many additional, and even including recently a few new therapies to help manage the condition, including to help prevent sickle cell crises, of course, infection prevention, directly treating anemia, and blood transfusions, for example, to prevent stroke. Some newer monitoring for stroke risk. Like I said, other additional new interventions. So this is an area where recently there has been a little bit more advanced in terms of research and therapies in principle available, although again, not even necessarily widely available in high resource settings and distinctly unavailable in lower resource settings. So genome editing as an approach for curative therapy, there are multiple approaches. So one is to add functional gene copies of the hemoglobin gene. Another is to directly, with gene editing, correct the mutated gene. And then the third approach is to increase fetal hemoglobin gene expression. Fetal hemoglobin begins prenatally and ramps down shortly after birth. But there are approaches to disable the downregulation of the expression of that gene, and thus restore expression of fetal hemoglobin at a level that that can rescue the phenotype. And so all of these methods are being pursued and are being pursued through different specific approaches. The two furthest advanced clinical approaches, one of which takes this gene addition approach, Bluebird, and then Vertex and CRISPR Therapeutics take this gene editing approach, specifically using non-homologous end joining to disrupt the regulators of the expression of the fetal hemoglobin gene. And then, of course, the way that this is delivered is with ex vivo gene editing. So, you know, just a quick diagram, I think people are pretty familiar, but of course, I think it's important to understand as we talk about these therapies, the burden of them. I mean, this is just an excerpt from a pretty recent New York Times article describing the patient experience of, 8 weeks of blood transfusion, and then, ultimately after when it's time to receive the autologous transplant, at that point they have busulfan or similar conditioning, and typically spend a month in the hospital recovering. So these ex vivo approaches are really impressive in many ways, but are a very intense patient experience and also very resource intensive in terms of healthcare resources. Still very exciting for sickle cell disease curative therapies, that there are two that are both seem to be on the cusp of FDA approval, and in one case probably also on a pathway to EMA approval. So for each of these approaches, clinical trial results have been promising in many ways, although there are certainly important open questions, including the long-term safety and efficacy. The FDA is requiring 15 years of follow-up, for example, in these studies. There are questions, as is referenced in the previous talk, for the Vertex and CRISPR Therapeutics approach, for example, of off-target editing or off-target cutting. And then also questions about patient eligibility and ultimately, of course, patient access. Beyond these, there are many additional companies and many additional candidates that are in the pipeline. Having said that, there have also been some notable exits recently for potentially varying different reasons, but one of which may be the fact that these two companies appear quite close to succeeding in registering their products. So, of course, there are many challenges anticipated for ex vivo cure. One is price or reimbursement. If you look at other curative genetic therapies that are currently available in the US, they're priced roughly in the $2.5 to $3.5 million per patient range. Estimates from a US perspective is that for sickle cell curative genetic therapies would be cost effective in a range that includes roughly $2 million per patient. So it is expected that there will be limited access, even in high resource settings. There will not be the possibility of this procedure in most hospitals, but it's also anticipated that in a country like the US, maybe 50 treatment centers would be sufficient to cover most of the potential population, in part because of relatively high concentration of the patient populations. Of course, I think it's clear if you go again to the area of the highest burden for sickle cell disease in the world, incredibly challenging to scale a therapy like this at this point in sub-Saharan Africa. Also, as I mentioned, there's these issues with patient acceptability, a very long, intensive timeline for this procedure, and it requires really high trust in the medical system. Again, particularly, for example, in the US context, there's a long history of justified mistrust in African American communities toward the healthcare system. And so this is another important challenge to scaling up an intervention like this. So, of course, that leads to the natural question of whether some of these challenges could be circumvented by shifting to an in vivo editing approach. Clearly, opportunities include lower cost and thus potentially, though not necessarily lower price, a much reduced infrastructure requirement, thus the ability potentially to scale it much more broadly globally. And of course, potentially much less demanding on the patient, thus making it potentially much more acceptable for patients to undergo this therapy, and potentially expanding the fraction of the sickle cell disease patient pool that could be eligible for it, because, for example, of not having to undergo conditioning. Having said that, of course, there are still many challenges, including potentially immune responses to the vectors delivering the machinery, achieving sufficient cell target specificity, and of course, editing efficiency. In terms of progress for in vivo editing for sickle cell disease, there has been demonstrated cures in mouse models, proof of concept targeting hematopoietic stem cells with both lipid nanoparticles and with viral vectors, including earlier this year, the demonstration of delivering a prime editor. So of course, there are companies working toward advancing in vivo approaches into clinical development. Again, perhaps in some cases, some evidence of a shift in that direction, perhaps motivated by the lead that some companies already have in terms of getting to the market with ex vivo cures. So looking ahead to next generation, actually both next generation in terms of the specific editing approaches and next generation potentially in terms of moving in vivo. I work at the Foundation for the NIH, so I do want to talk a little bit about the NIH's work here. Sickle cell disease generally is a priority for NIH. It invests about $100 million a year in sickle cell disease research. And sickle cell disease cure beyond that is a particular priority. This research is led out of the National Heart, Lung, and Blood Institute. The NIH has both intramural research and makes grants, of course, to advance sickle cell disease cure. One role in particular that FNIH plays, we have funding from the Bill and Melinda Gates Foundation to support intramural research on engineering LNPs and lentiviral vectors to specifically and efficiently edit hematopoietic stem cells, exactly the kind of work that we were talking about on the previous slide. Also, I wanted to mention the NHLBI leads a partnership called the Cure Sickle Cell Initiative. It involves several US government agencies, many grantees, and one of the very important aspects of it, coming back a little bit to Luis's introduction, talking about the voice of patients and the voice of caregivers and their importance in this space. So this partnership has a community panel that includes patients, caregivers, and advocates that helps steer the direction of the program overall and the funding, and serves as a resource for partners in this. And certainly including patients, advocates, caregivers in this kind of research is incredibly important for many different reasons, especially, of course, including ultimately, delivering confidence in the products that come out of this research for the communities that would most benefit from them. And then finally, just a brief comment outside of sickle cell disease. So this headline was from 2019 of the NIH launching a new collaboration, a collaboration with the Bill and Melinda Gates Foundation, looking at the intersection in particular of gene-based curative strategies for sickle cell disease and HIV. So HIV, almost a 40 million population of almost 40 million people that are living with HIV worldwide. So of course, the ultimate potential of achieving some kind of functional cure for that population, tremendously important public health goal globally. And so it is recognized that although unlike sickle cell disease, it's not yet, well, scalable genome editing approaches for curing HIV are not yet understood. It is expected that at least some of the elements that could fall into some of those approaches might share some overlap with some of what is already being developed for sickle cell disease. So there's a deliberate effort to bring those efforts together and allow them to leverage common platforms where they can. And so with that, we'll be happy to wrap up, I guess, a couple of minutes early. Thank you very much.
Thank you very much, Michael, for this interesting presentation. Luis, do you see some questions on these three presentations? Luis, can you activate your microphone, please?
Yes, yes,
Please.
thank you. There were a couple of questions that were nicely already dealt with by Alta Shadow, so thank you, Alta. And while we're waiting for other questions to come, I have questions for the three of our speakers, including I don't see Andre. Andres.
Right.
Yeah, actually my video is
okay, so I will be reactivating your video. Yes. Okay, now you can reactivate your video. Okay, thank you.
Thanks.
So, Andre, it looks to me that your pioneer use of talents has provided you with more specificity, because at the end of the day, as you said, there are two elements and you are matching longer stretches of DNA. So this is like an impression, and this is the fact that you're seeing when you're analyzing the specificity of your treatments.
Well, there are like multiple elements. First of all, usually we don't have any base pair constraints such as like a three base pair motif such as a PAM. So you can select exactly the place where you want to cut. So usually the more you have, there is a need of a motif, the more you have difficulties to find exactly the place where you want to cut. And the second thing is that I think is a great advantage is that once you design your protein, as it's, you know, an RVD DNA interaction, so there is like 2 amino acids per RVD that interacts with DNA, you can continue to re-engineer the protein, like the TALEN, up to the time you have a very high specificity versus activity. So generating a TALEN takes like less than a week. It's like you can do one like this like in 5 days. But finessing this up to a therapeutic grade takes like a couple of months. And however, the product that you get at the end is extremely powerful. It's of course like at the base pair, and the DNA interaction is extremely high. What I think personally is like really interesting and that has never been envisaged is the DNA, like the DNA ends release. Usually when you release the DNA ends, and I like spoke about it very rapidly, it has to be done with the same speed on both sides, because one of the first things that the cell will do is like send exonucleases 5' to 3' to unzip DNA. And these are the things that are interacting and will do are reactive, like autoreactive for DNA recombination. So the more you release very quickly at the same time, the less you will have one that is blocked by, for example, cats that have a KD that is super high on one side versus very low on the other. And you have like only one reactive end hanging out in the nucleus and have like unbalanced recombination. But all in all, you know, like we're totally technology agnostic on our side. We've used CRISPR, we use TALEN, we use meganucleases, zinc finger nucleases, etc. At the end, it's of course whatever works the best. So far, we've been thinking that this technology have like shown a lot of robustness on this side. That's why we stick to it.
Thank you, Andre. Denise, I loved your approach using this RMP, this naked approach. I guess for eye treatments, being the eye an immunoprivileged organ, it's okay. What about other organs? Wouldn't you be concerned with the immunogenicity of these Cas9s and the existence in most of us of antibodies against the standard Spiogenes and Staphyloaureus Cas9-like proteins?
Yes, I would be very much concerned with both of those things that you've cited if I were doing a systemic kind of delivery approach. I'm just enjoying the eye because we do have this privilege. We're away from systemic effects, and we actually have an opportunity to be at the forefront and do things that are a little bit more bold. Same for inner ear or brain. Yes, I think I have less constraints. But immunogenicity is still a problem. And in fact, the preliminary studies that we've done point to the guide RNA being more the culprit of the immunogenicity rather than the protein part. So I think we have ways to go to work on that also. And generally speaking, if we are able to find these vectors that can, so with the naked RNA, we can already obtain a good amount of editing, up to 10%, if we're targeting the right gene with the right guide RNA. So if we can improve that by tenfold, let's say, and reduce the dose, that's still going to allow us to improve and reduce the immunogenicity from that point of view. And second, if we are able to shield these with any kind of peptide or lipid, that might also go a little bit in favor of not getting in trouble with the immune system. So those are future perspectives that we need to implement.
Thank you, Denise, and congratulations for your work. Michael, my question for you is regarding sickle cell disease. And being this the disease that probably will be benefiting from the first approval, early this year, we read a report from colleagues in the Boston area that they found out that there was a genetic variant in people from Afro-American origin that was kind of a perfect match for one of the guides that is being used in this gamma reactivation treatment, so in this BCL1A. And this wasn't found before because the genomes that were used for look for off targets did not include enough amount of Afro-Americans, which already speaks about our biased way of analyzing off targets and speaks in favor of trying to use pan-genome approach or at least multiple genome approach. Do you think this might interfere or delay the approval of this approach?
Delay the, well, I mean, I don't work for the FDA, so I probably shouldn't comment exactly, but I think the impression is still that these therapies are on their way toward approval based on the advisory committee results in one case, in the pathway in the other case. Not that that's not consequential, but my guess is given the discovery that it's just going to affect the eligible patient population.
Well, bad luck in this case is that this second match is on the same chromosome, so this is even favoring inversions and aneuploidies. So this is probably the worst that you can expect. Anyway, so we will have to look. Francois,
yeah.
I see.
Yeah, we have several questions.
I see several questions.
Yeah.
Do you want to read it? Read it yourself?
Yeah. That was about to ask the question to Denise. So Joseph Kamgono asked this question. Thanks for the great presentations on genetic therapy specific to retinal dystrophy. Could other problems such as diabetic retinopathy or blindness due to glaucoma could find a solution?
Yes, I was replying by chat,
okay.
but I can also, of course, voice what I just said. So yes, gene editing is already being implemented for glaucoma and diabetic retinopathy because these tools are extremely versatile. They will find applications, I think, all across the board. Also in complex diseases as the ones that you are citing. There are preliminary examples of them that have been done in laboratory animals. I will put in the link a review to to what I was referring to. So you can see where at which stages they're at. And you can find that there are early examples which hopefully will slowly build towards clinical applications in the years to come.
Okay. So there are other questions from a colleague, Issa Toussa, from Banjul, I think from Gambia. So the in vivo use of CRISPR-Cas system has faced many challenges, such as the possibility of causing autoimmune disease, the identification of a proper delivery technique, and ethical concerns. Sorry. Do you think that the use of AI machine learning may help to overcome these challenges of autoimmune disease and other issues? So could answer to
I guess this can be answered by any of you. So please feel free. Andre or Michael or Denise.
Well, it's very
We
difficult.
can't avoid the question of AI, right? It's impossible to.
I can say a few words, yeah, like, well, AI has been like, AI is a very cool term for, you know, algorithms. Algorithms has been a center part of all the current development in biology, molecular biology in general, and of course in gene editing. And I think this is going to expand even more in the future. I think that there's a lot of solution or helps could come from there. So I guess that yes, the answer is yes. Now it's like very difficult to guess what's going to happen in the future, because like we can make educated guess, but if you want to translate what has been the revolution for the past 30 years in biology, it's definitely not biology, it's robotics and like computer sciences, algorithms. Like with robotics and algorithms, like most of the big challenges such as sequencing, like genomics, protein folding, etc., has been solved. So then we're just helping this to happen. So like the answer is yes.
Okay. Denise, you want to say something?
Sure. I mean, yes, like exactly as emphasized, I think AI is finding applications everywhere. So within, for example, I can just give an example in the delivery perspective. So for example, we're trying to develop these mechanisms that are going to help the RNPs enter the photoreceptor or RPE cells better. And what you need for AI to work is a set of data where you've tried to solve a problem. So for example, if you do a combinatorial experiment trying to choose peptides that help targeting a specific type of cell, then AI can be used to learn from that data set and propose even more efficient versions of those peptides that would help you specifically target a cell and gain entry into it. So that's one example, for example, how AI can be aided in designing better delivery vehicles. And we are using this type of approach to design peptides, for example.
Okay, thank you. So before addressing the social issues linked to these progresses, another question from RC Toussa: are we ready to use CRISPR-Cas gene editing in clinical settings to target cancer-causing genes? So Andre, you want to comment on that?
Well, I think the approach of using cell therapy for now have a lot of to go. Well, I think that the use of gene editors such as CRISPR-Cas just to target the genes that are causing cancer or trying to fix them might be a bit challenging in the very short period of time. It might be something that could be fixed in the future. But for now, essentially, well, we face essentially the limitation of most of the gene editing and gene therapies, which is like vectorization. So it's better to try to treat the result of these mutations in trying to clean up using cell therapies such as CAR T therapies or pills or tumor-infiltrating lymphocytes or other type of approaches that could come as a second shot, because this could be pretty widespread in terms of expression. So not in the immediate time.
Okay, and now to follow on Michael Santos mentioning the equity of access to these technologies, there's a question from David Curie. A recent Lancet Commission hematology on sickle cell disease suggested a 2040 horizon before meaningful, affordable gene technology access. Do you think that it's aggressive enough to look for 2040 as an horizon? And also, you are developers, Denise or Andre, do you foresee some potential transfer of technology to the low and middle countries, or?
I can say a few words about this. Actually, it's like, for example, for sickle cell disease, the cost of one patient having sickle cell disease for the whole healthcare system in Western countries in general, like essentially United States and like Big Five in Europe, is like pretty high. And bringing a therapy, first of all, that can be extremely relieving for the patient and that don't have to spend period past of his time in having transfusion and other type of treatment in the hospital. So for the comfort of the patient. And then finally, for the healthcare system, I think gene therapies, especially gene editing, treating the root of the disease versus trying just to simplify the symptoms in general, will reduce tremendously the cost of the patient at the end. Don't have to look at the cost of the therapy, even if it costs, for example, $1 million or $2 or $3 million at the end. For over the lifespan of the patient, if the patient receives a curative therapy, then I think at the end, the healthcare system will be a winner. And the decrease of the healthcare system would be massive using gene therapies and gene editing in general. That's what we should see, not the cost of the therapy itself.
Denise, given the price of Luxturna, for instance, this therapy, which was absolutely unaffordable for many countries, how do you see the future?
Well, I see this as a very dark picture. I certainly don't think that 2040 is aggressive enough. I think we can do much more in the way of this, but I think it's outside of the scope of the kind of investigative scientific work that we do. This is more of a political effort that needs to be done. And I think this is true for not just gene editing, but all innovative therapies, because they require a lot of investment in the early stages. Private companies justify their cost by referring to the cost that it took to develop them. But I think we need to have a more long-term view into these and clearly keep this ethical aspect in front of our eyes, not to lose sight, and develop funding mechanisms and ways that allow to make this access possible.
But do you already collaborate with research team in these countries, or so you have you only foresee the transfer of medicine? Or
so that's an excellent question. No, I don't. What we're doing is more acting at the local level, trying to see already, because these are more rare diseases already in France and within the region, Parisian region. We're trying to put together a pipeline and hotspots for the regulators to agree on, which would lower the cost for us, first of all. And then I think we can reach out once we have it figured out, once we have a clear scheme, because right now, even people working on this kind of gene therapy for rare diseases for different tissues are not collaborating enough that the efforts can be federated in towards the aim of reducing the developing cost. And then there's also all of the clinical aspects, regulatory aspects that also need to be addressed. First of all, within us here, who are at the forefront, and we need to have a consensus on that and federate that effort to lower the cost. And then we can think about transfer more efficiently. So I think that's going to come of age soon. But currently, I think we need to work it out, first of all, where we're at.
Thank you. Luis, do you raise your hand?
Yes, I remember Alta and Jennifer and myself were attending in March the 3rd summit on human genome editing in London. And I do remember Victoria Gray, the first patient being cured by sickle cell disease treatment with CRISPR, being so happy to present there. But also I do remember the patient representatives from India, Tanzania, and South Africa with very disappointing faces, because they did realize that even though what Andres said is absolutely correct, and it is legitimate to expect that this price that needs to be paid might compensate, even be less expensive than the whole life treatment. The reality is that we need to find a solution that combines the legitimate access to generating the profits for the company, combined with a reasonable price that could be paid for national health systems. Otherwise, this would be treatment for the rich, and this will not be treatment for, remember the map that Michael has shown us. Most of the patients are in Central Africa, in Sub-Saharan Africa, and we need to reach those. They do need to be treated as well. Alta wants to say something.
Yeah, sorry, Jennifer, we started the second session. If you want to take the lead. No, please.
Yeah, thank you. And just to follow on with what Luis was saying, one thing that our community has not yet done is to really examine some of the business problems associated with the development of gene therapies and why they are coming out at the price point they are. Some of it is technological. Some of it is the regulatory pathway. Some of it is logistics, the transportation and storage issues that are more complicated for these things than for other kinds of traditional drugs. But some of it is simply that we have a business model that is premised on much investment by companies, many failures, and then the occasional success for which they then get a return on investment that covers all of their losses and their profits and their future investment, without some kind of national system to amortize those costs the way we do when we finance houses. And as a result, we lack an incentive system that would properly, as Luis was alluding, properly reward companies for investing in high-risk research without requiring that it be handled entirely by the upfront cost as soon as a therapy is approved. And so until we have a truly different business model for incentivizing investment or compensating for losses, we're not going to solve this problem.
Francois, there is 1 minute left if you want to read an additional question.
I lost the question, sorry.
So there is a question from Misatu Sar as well. I'll be reading for you. So for some disorders, gene therapy will work only if we can deliver a normal gene to a large number of cells, and once it's turned on, it must remain on. Cells have an habit of shutting down genes that are too active for exhibiting other unusual behaviors. Also, introducing changes into the wrong cells, targeting a gene to the correct cells is crucial to the success of any gene therapy treatment. Just as important, though, is making sure that the gene is not incorporated into the wrong cells. Delivering a gene to the wrong tissue would be inefficient, and it could cause health problems for the patient. It was a comment rather than a question. And I think we can leave it like this. I think it's 20 minutes past, so Francois, I think we can move. And,
just a comment on the last discussion from Malta. I did a review for a journal on the use of monoclonal antibodies in therapeutics throughout the world. And I found out that despite the high cost of the use of monoclonal antibodies, many countries now developed some local capacities. It was in Brazil for years, but also in India. I found out in some African countries as well to have local capacities to develop monoclonal antibodies for the local use. So it's possible in 2040 perhaps, or perhaps beyond for the use of genome editing technology. So, having said that, I hand over to Jennifer. We started this, as I said, we started a social discussion. So, sorry, but
let's thank Michael, Denise, and Andrea. Thank you.
Our pleasure. Thank you.
So Jennifer,
of course, you can stay if you want, of course.
Of course you can stay, please. Thank you, Luis. I don't see Catherine Littler. I don't think she's joined us yet.
He said she joined very late because she was
all right. So
you can carry on with
so she would be the third. Yeah, she would be
all
the
right.
third.
So I will carry on, and I think we'll start with Alta because she's going to be giving us a sort of an overview, a panorama, a survey of the social and ethical issues related to certain practices or certain proposed practices of germline editing. So just a few words about Alta, who needs no introduction, but I'm still going to make one. Alta is Professor Emerita of Law and Bioethics at the University of Wisconsin, and now she's working as a consultant to biotechnology companies. And some disclosure here, some have genome editing technologies and therapies in development. She co-chaired the U.S. National Academy's studies on stem cell research guidelines and on genome editing policy, and I had the huge pleasure of working with her following the first summit in the working group that produced the report that was published in 2017, excuse me, and also was with Alta and some other members of ARRIGE, including Albrecht Müller Seminar in Hong Kong, when Dr. He just sort of blew the roof open with his announcement. And I think she's going to say a few things about that. Alta served also on the WHO Genome Editing Expert Panel, which Catherine directed and on which participated as well Albrecht Müller, on the organizing committees for the International Genome Editing Summits, the executive committees for drafting the 2022 International Society for Stem Cell Research Guidelines for research and clinical use of stem cells, including genome editing. And she is now working with Tippy McKenzie at the University of California, San Francisco on draft consensus standards for in utero somatic cell fetal editing. And I think that's going to be something she's going to talk about next week when I get the pleasure of seeing her live in Paris for the INSERM annual day. So Alta, I turn the floor over to you. And as with Francois, we'll hear from our three speakers and save the questions for last. So you might want to start writing them in the chat box and I'll survey them. And when I get the floor back, I will address these questions to each of our speakers. Thank you, Alta.
Thank you very much, Jennifer. And let me just ask, is it okay? My slides, my audio,
Yep.
is that all working?
Everything is okay,
Great.
Alta. Go ahead.
Great, thank you very much. Yes, as Jennifer said, I'm privileged to just offer a very brief survey of what's going on in the world in terms of policies and progress here. And just to follow up first on that conflict and bias disclosure, I think Jennifer's listed pretty much everything, but at the very bottom of this slide, you can see the names of the companies and organizations. I don't plan to talk about any of their products or ongoing research in this presentation. The following information on somatic editing is taken from the Genetic Literacy Project Regulation Tracker, so I'm hoping it's fairly up to date. Somatic editing, which we've been hearing a lot about for the last hour and a half, is something that is subject in most highly developed countries with highly developed regulatory systems to the same level of rigor as we're used to seeing for other therapeutic interventions. The other thing that's interesting, and you see this both in the European Union and in North America, in the US, or even in Brazil, that the approach has not been to write special rules solely for gene therapy, but instead to be looking at the rules that have applied overall to the regulation of biologics and see how far they can go in the regulatory concerns that are raised, particularly with gene therapy and with gene editing, especially. Those concerns, as you've already been listening, already hearing, sometimes have to do with some of the special concerns about long-term problems due to the delivery systems with immunological reaction and the inability to then follow up with second or third doses if those are needed. Another concern has to do with the off-target effects in the area of gene editing. And raised in the question and answer column here in Zoom was the question about follow-up and the need for long-term follow-up. In the United States, that was the subject of a guidance going back to 2020, suggesting 15 years as a minimum. And that in the Q&A, I actually listed the details of that Federal Register notice in the US, talked about some of the special concerns about latency periods and such that have triggered that particular requirement. It's a complicated requirement because in this field, some companies don't last 15 years. And by the time that follow-up period has expired, the investigators and even the company may no longer be present. And so special attention has to be paid to how one constructs a long-term follow-up plan and puts into place financing at the front end to ensure that that follow-up can continue. But the bottom line is that in the area of somatic editing, we do have regulatory agencies in place. They are still working to figure out how to measure off-target effects, how to evaluate the significance of off-target effects. And so for the moment, there is a rather slow pace in the review process. But as was mentioned earlier, we've seen in the US the first sickle cell therapy go at least as far as the advisory committee, and despite some questions, a generally positive response based on the risk-benefit analysis. You can see here other countries similarly in the UK, Australia, New Zealand, that these are all highly regulated, but the research is proceeding. By contrast, there are some countries where the regulatory process is a little bit less rigorous. I think that here in Israel, we actually do have a sophisticated regulatory system, but there is a somewhat more aggressive approach to moving gene therapy forward. And in Russia, it's actually, I have to confess, a little bit hard for me to completely be sure about the information, but it does seem that they do have a Ministry of Health that does oversee this, with the need for a special approval by central and local ethics committees. But it's unclear to me exactly how far they've progressed in the detailed regulatory detailed review process for things like off-target effects. China is a special case because China's been going through a set of revisions in its bioethics laws and its research oversight rules. But at this point, that revision process is not yet complete. It was spurred in part by the events we'll talk about in just a moment with Hu Zhongqui's editing of human embryos. But it's against a backdrop generally in China of a very complicated system with some national guidance, but a great deal of decentralized oversight by research committees at the local level with widely differing levels of expertise and widely differing levels of understanding even of the laws that apply, according to some of my Chinese colleagues. More recently, there have been some efforts not only to rationalize and to make more rigorous their overall research ethics oversight processes, but also, and most recently, an effort to extend the bioethics reviews and the ethics reviews generally beyond just government-sponsored research into non-government, privately sponsored research. And for this information, I thank my colleague Joy Zhang at the University of Kent in Canterbury, UK. With human trials, you've just been listening to some of the most exciting work that's being developed. This was a slide I prepared before I realized how good the early presentations would be. But again, it just reviews for you the wide variety of areas in which there already are human trials going on or just starting. And so we're seeing an explosion of research in the area of somatic editing. And as we were just talking about with sickle cell, in the United States, we now have the first sickle cell anemia lifelong cure that is heading toward an approval, I predict. But we also have some cautionary tales. And here we have the example of a patient with Duchenne's muscular dystrophy, whose death may very well have been due simply to the high dose of the AAV vector, but it is still something that is a cause of great concern, is how to identify the right dosing. And for this, I'm now going to simply defer to my medical and scientific colleagues to go further. One area that is not discussed very much, but Jennifer alluded to it in her very kind introduction, is editing not the embryo, but editing the fetus. So here are cases of people who get pregnant, and then during the course of the pregnancy, usually sometime around the beginning of the second trimester, they discover that there is a significant medical problem that will affect the fetus and either cause fetal demise or will cause the live-born child to have a very serious illness or disability. Some examples have to do with Gaucher disease, spinal muscular atrophy, Leber congenital amaurosis. These are all the kinds of disorders that are now being picked up at the fetal stages. And one question was, can we do something about dealing with this disease before a child is born. There are some specific advantages to working in utero for certain diseases. For one thing, the fetal immune system is really wired for tolerance, and it means that there may be a lack of immune memory that handles this redosing problem. That is, you can dose at the fetal stage, and if need be, once the child is born, it's possible this means the child could be redosed if the gene therapy is no longer is wearing off, essentially, which we have seen in the area of Duchenne's muscular dystrophy. The second is that it might be easier to access the hematopoietic stem cells in the fetal liver, and therefore be able to get to them, edit them, return them before they've retired into the bone marrow. That creates opportunities that may not otherwise exist as easily after birth. The permeability of the blood-brain barrier allows for a different level of access to the central nervous system. The size of the fetus, just the size of the fetus, allows for lower doses, and that itself may be an advantage in terms of safety. And it also means that manufacturing costs may be less. The other thing that's interesting is that when you take, I think I must have deleted that slide. Another thing that's interesting is that in some cases, you can get access to tissues that need to be edited more easily. So, for example, in cases of things like cystic fibrosis, we've discovered that getting the delivery to the deepest parts of the lung can be very difficult in adults or children. But for things like cystic fibrosis, it might be easier in the fetal context to access those tissues and get the edits in place. So overall, there are these advantages to doing editing in utero. Obviously, as soon as you start talking about doing interventions during the course of pregnancy, you have a number of additional complications. One is that you're dealing with two patients because you have the pregnant woman as well as the fetus, and you have to look at whether or not this particular approach offers any risk to the pregnant woman herself. The second is that there's a second parent, and at least in the United States, our rules require that that second parent, usually a father, but not always, is consulted and has some role in deciding whether or not the research can go forward. Third, at least in the case of the US and some other countries with complicated politics around abortion, Poland is an example these days, you find that we are now looking at women in positions where they no longer have the option to terminate a pregnancy when they have a fetus with these problems. And so in that sense, they may be driven to accept the offer to engage in these very experimental therapies in a way that before they wouldn't feel pressured to do. They would have the option of simply terminating the pregnancy and trying again in the future. In addition, at least in the US these days, the politics around the change in abortion rules and the restrictions on abortion have led to state laws being passed that are so confusing to physicians that they are no longer even handling complicated pregnancies the way they would like to. They are scared to terminate pregnancies or to treat women for conditions where it might affect fetal health because they're unsure what the state law actually requires of them. And so we are now, at least in the US, facing a situation in which the restrictions on abortion may be actually making it harder to do fetal health and child health promoting research like somatic editing in utero. I do want to say that everything I know about in utero editing really does come from my colleague, Tippy McKenzie at the University of California, San Francisco. On heritable germline editing, there really isn't a lot to say here because it is still illegal in countries that are signatory to the Oviedo Convention. In the United States, it is still impossible to do it legally, not because we actually have a law that says you may not do germline editing, but because the US Congress has a budgetary constraint that it has placed on our Food and Drug Administration, which must give permission before any clinical trial could begin. That budgetary constraint says that the FDA cannot even acknowledge, let alone to accept and review an application to begin a clinical trial that would involve heritable germline editing. So the result is that since you need the FDA's permission, but the FDA is not allowed to give it, it is functionally illegal, even though it's not formally illegal in the US. That said, the discussion around whether or not it could ever be done has continued. There was a report by the International Commission, which was organized by various national academies of medicine and national academies of science, the US and UK among them. And that report actually did posit the possibility of germline editing in the future, similar to the report that Jennifer and I served on for the National Academies back in 2017. Similarly, it outlines extremely narrow indications that might meet the kind of compelling need argument that is needed to justify this kind of rather dramatic intervention. The International Commission also went into great detail about the other kinds of preconditions that have not even that have not been met and are not even close to being met at this time. That includes extensive preclinical, that is non-human research, both purely in vitro as well as non-human animal research on both safety and efficacy, the rigor of the regulatory oversight, the kind of long-term follow-up that would be needed. And then the International Commission also had suggested that there ought to be some kind of international committee or committees looking both at the ongoing scientific progress on this preclinical work as well as on the ongoing transnational discussions about its social acceptability. At the March 3rd International Summit on Genome Editing in London that Luis has referenced several times now, the organizing committee, and keep in mind the organizing committee does not represent any of the national academies, but it was a group that had organized the summit, agreed on the following statement, that it should not, that this kind of heritable editing should not be used unless at a minimum it meets reasonable standards for safety and efficacy, is legally sanctioned, and has been developed and tested under rigorous oversight. You'll notice that if you look at the membership of that organizing committee, it included people who were adamantly opposed to germline editing at any point or any time, and those who think that it is defensible, even though it is absolutely not ready yet, nor has the compelling case been made for it yet. And yet that group with these differing opinions was able to agree with this particular statement. Notice also the comment about legally sanctioned is there deliberately to acknowledge that there will be global diversity in approaches on this topic, with some countries continuing to make it illegal no matter how compellingly people argue for it, and others might in the future approve it if it meets other standards. People have been asking what happened to He Jiankui. So as you know, he was the person who announced at the second summit that he'd edited embryos resulting in multiple pregnancies. At the time, there'd been 2 births, since there should have been 1 more. I do believe he genuinely expected that he was going to be lauded as almost a kind of, Nobel Prize winning scientist who had set the stage for a new era. Instead, he was widely denounced. And if one was there and looked at the data that he presented, you can see that he didn't actually achieve the edits he claimed to be editing, let alone had any compelling need. He was doing this for couples that were discordant on HIV status, where it was the father who was positive, not the mother. That alone tells us that this was nothing, there was no argument for this, since the chance of transmission was incredibly low and sperm washing offered an alternative way of reducing that already low risk even further. Nonetheless, he persisted in doing this. Subsequently, he was subject to house arrest and then was convicted at the time for practicing medicine without a license, because the Chinese law at the time did not clearly prohibit what he had done. They've been working on changing that since then. There have been a lot of questions about the status and health of the children who were conceived this way, and that information has not been forthcoming, the argument being made that they deserve to have their own privacy protected, and that is certainly true, but at the same time, I think there's also a legitimate desire to know how they are doing and what the effect might be of some of the unexpected targets that were hit in his editing experiment. He Jiankui most recently has been back in the news. First, he was invited to give a presentation to Chinese authorities and ethicists, at which time he refused to answer even the simplest questions about his current plans. Since then, in just the last few months, he's been out again announcing that he wants to try germline editing, this time not for HIV prevention, which again was not a justifiable kind of compelling use, but now for the prevention of Alzheimer's disease. Again, not a compelling purpose, since we're talking about a disorder that doesn't usually hit for many, many decades, meaning that there are many opportunities for the research community to tackle that disease long before people get it. And so what he's talking about is, again, something that is not easy to justify, nor has the preclinical work been done, nor does he seem to have the technical capacity to do it responsibly. But until he does it, it's not clear to me he is violating any laws. So for the moment, he's more a publicity stunt than he is an actual threat. With that, and I unfortunately couldn't even see the clock while I was doing this because it took up my entire screen, I'm going to stop and turn this back over to Jennifer. Thank you very much. Jennifer, you're still muted.
Sorry. I was saying that you had 1 minute left, so you did great. Okay, lots of questions, some in the chat. I have some as well, but we're going to move ahead and hear from, excuse me, from Dr. Thalder, who is present. And Dr. Thalder is a full professor of law at the University of KwaZulu-Natal in Durban. He has published over 80 articles in local and international academic journals, and is the principal investigator of a research project on the legal aspects and the use of data science in health innovation in Africa, excuse me, which is funded by the NIH. He also has a private legal practice and has served as a legal counsel in several reported cases, and these include those in the Constitutional Court, South Africa's Apex Court. And I think today, Dr. Thalder, you're going to be talking to us about work that you carried out in South Africa.
Greetings, everyone. I trust you can see my slides.
Yes.
Excellent. So the topic of my talk today is the not-so-sacred human genome, analyzing the results of a deliberative public engagement study on heritable human genome editing among South Africans. So to start off with, I think in various publications related to genome editing, there's always an emphasis on the importance of public engagement. So my research group decided that we should take that seriously and do a public engagement study in South Africa, which was then the first one of its kind in Africa. We decided on the deliberative public engagement methodology because that offered two specific advantages for us. I think the first big advantage of the deliberative methodology was that we could ensure that all our participants were very well informed about what the technology entailed. And we accomplished that through, when we advertised through social media to recruit participants. After they've registered on our website, they were provided access with background information, about 12 pages that informed them about the technology itself. In some cases, it referred to some videos available on YouTube, and it also provided them with a balanced overview of the various ethical positions on heritable human genome editing. This training material or information was also peer-reviewed by some international peer reviewers to ensure that it is or that it was as balanced as possible. So the first benefit that we saw to the deliberative methodology was the fact that we could really ensure that our participants were informed. They had to study this material, and after that they had to to sit an entrance exam of 20 marks, and they had to get 100% in this entrance exam that they could do online before they were included in the pool of potential participants. They could, of course, redo this exam, but they weren't given any indication of which question they had right or wrong. So eventually, I'll share the numbers with you soon. So that was the first benefit. We had informed participants. And then the second big benefit was it wasn't just their original positions or their knee-jerk reactions, but this whole public engagement was facilitated in a consensus-seeking manner. Not that consensus was ever enforced, but it was facilitated in a consensus-seeking manner to promote more deliberation and interaction between the participants. So the results that we got were also the results after such deliberation. We consider that this was very much aligned with South Africa's political culture of participatory democracy. So in line with the literature, we decided that 30 participants would be optimal to allow for this kind of discussion. We sometimes use all 30 in a plenary session, and then sometimes broke them up into smaller groups of 5 or 6. And once we had our pool of potential participants, we also very carefully compose our group of 30 to be as inclusive as possible on various demographics. So that's the number of people who were interested and registered on our website. We were very pleasantly surprised by all the interest. However, only 70 of those people eventually passed the exam with 100%. Some of them took up to 10 times, retook the exam up to 10 times. They were clearly very motivated to be part of this. And from then, of course, we selected 30 to participate. And regarding that, one of the demographics was also to ensure that there was a proper spread of educational level within our eventual participants. So because this took place in 2021, it was still during the time of the pandemic, so it was done virtually over 3 evenings. So the 3 evenings each had its own theme. The first theme was using heritable human genome editing for prevention. The second one was for immunity against certain diseases. And the third evening's theme was enhancement. So, ladies and gentlemen, I will now take you through the attitudes that were expressed by our participants. So at the end of each evening, our participants voted, and then also a week later we asked them to vote again, and it's that eventual last vote a week after the deliberations, perhaps after a bit of a cooling period, that I'm presenting here. First, well, I will just first make some general comments before I take you through the specific voting on specific issues. So a general observation was that there was no principled objection against human heritable genome editing. The main paradigm of the discussions was more a benefit-risk analysis, a typical health benefit to health risk analysis, where our participants in debating each other would consider the seriousness of the condition and the potential benefit that heritable human genome editing could potentially provide, and then compare that to the potential risk that can also be involved. And another interesting theme was also the idea of equal access to this technology. The idea is that if it's seen as beneficial, then government must go out of its way to ensure that there's universal access to such a beneficial technology. In fact, some of our participants specifically mentioned that the South African government should actively invest in research on using heritable human genome editing for health purposes, instead of just waiting for the global north to do the research. The paradigm changed a little bit in the third evening that looked at enhancement. So there the paradigm was more about the perceived procreative autonomy of prospective parents versus ideas of diversity, and also the perceived autonomy of prospective children. So let me now go into the detail of the voting results. So within each theme, there were a number of questions, 4 or 5 or so specific questions, but all the questions started with the following phrase: provided that it is safe and effective, our country's laws should allow parents to choose to use genome editing before a child's birth to, and then the specific application, and the voting results. People could either vote unconditionally yes or no, and then there's a middle category: yes, it can be allowed, but subject to certain conditions. And I will talk about those conditions where relevant. So here is the first question: provided that it is safe and effective, our country's laws should allow parents to choose to use genome editing before a child's birth to prevent the child being born with a serious heritable disease such as sickle-cell anemia, muscular dystrophy, or Alzheimer's. And there, colleagues, you will see that it is overwhelmingly an unconditional yes. I then proceed to the next one. The next application of HHGE was to prevent the child being born with a less serious heritable disease, such as asthma or eczema. So still an overwhelming support, overwhelming yes, but a majority of people with conditions, and those conditions that one can clearly infer from the discussions were, well, the main one was the issue of equal access. The idea that if this is going to be a burden on the public purse to make this available, it is essential that there must first be a focus on the serious diseases, and that the limited public resources must first be focused on ensuring universal access for everybody on the previous question, on access to the technology in the context of serious diseases, and only then should it then move to the less serious diseases. So here we have the third application: prevent the child being born with a disability like deafness or blindness. And again, a rather overwhelming unconditional yes. The next one is prevent the child being born with albinism. So again, large majority yes. Now the conditions here are somewhat different. The conditions here focus a lot on the stigma and the discrimination attached to albinism in contemporary society, unfortunately. The idea here was that there should be specific educational and awareness campaigns by government if this is allowed to fight such stigma and discrimination, so that the technology that is then used to prevent this shouldn't then be seen as a vindication or something of the current discrimination against people who are actually born with albinism or living with albinism. The next one is prevent the child being born with Down syndrome, and there an overwhelming unconditional yes that heritable human genome editing should be allowed for this. I should also add for some context that NIPT, that is mainly used to detect Downs, is commonly available in South Africa, although it comes with a price tag that most probably limits its accessibility to the middle class. But it's not something that is unknown. Good. So now we're moving on to the second evening that focused on immunity, how this technology, how heritable human genome editing can be used to make future people immune against certain diseases. So the first disease was TB. And here, for some background, I can mention that both tuberculosis and HIV are currently still epidemics in South Africa. So it's not a peripheral kind of disease as it is in the global north. This is unfortunately quite common in South Africa, and TB, in fact, is the leading cause of death in South Africa. So it is quite serious. So that's the context in South Africa. That is quite different, I think, from many other countries, especially in the global north. So this is now about immunity. I'll just read this. Make the child immune to contracting a serious disease like TB during the child's life. And as you will see, overwhelming support, most of it also unconditional. A portion of that was conditional. Again, there the attitude was, again, that public resources should first go to the serious heritable disorder, disorders mentioned in the first evening. Let's go on to the next one. That is HIV/AIDS. So with this one, there was also a discussion, but to an extent at least, one can view contracting HIV as a consequence of choices made in one's life. But we all know that despite a whole generation, despite 20 years of attempts to educate and create awareness, HIV/AIDS is still an epidemic in South Africa. So clearly everything that our government has done with the massive international support as well over the past 20 years did not work to get rid of this epidemic. That can, of course, contribute to how people feel about this. So again, you see the results here. Do note, of course, that one should clearly differentiate this result from the Chinese scientist, Professor He, and his experiments, given that our question was clearly prefaced by the statement, provided that it is safe and effective. So that would then suppose preclinical and proper clinical trials. Good. The next one is make the child immune to contracting COVID-19 during the child's life. And here, colleagues, you can see the overwhelming support that we had in the previous cases now being broken down. It's now almost even. Still a majority, yes, support at that stage, at least during the lockdowns, but you can now see the opposition mounting. And the first one where there's actually then a slight majority against would be this one, to make the child immune to contracting an illness like the flu or a common cold during the child's life. So here we move over to the third theme of enhancements. And clearly you will see that the opposition is now in a majority. Here it's about using heritable human genome editing to influence talents, such as how intelligent or athletic the child will be. What is interesting is that there is still a substantial minority that would be in support of this. And of course, the argument was complex, but something interesting that was raised was that there can be general societal benefit to having a more intelligent population. Just an interesting comment that seemed to have influenced people during the debate. But as we then go on to other instances, the opposition starts increasing. So here we have influenced personality traits, such as how aggressive or cooperative the child will be. And remember, I mentioned here, instead of being a cost-benefit analysis, a health cost-benefit analysis that was the predominant paradigm during the first two evenings, here it was more about, now it's purely a matter of the parent's autonomy to decide versus values such as diversity and values such as the child's own autonomy to decide on certain things. Especially here, with this question about sexual orientation, the issue about the child's autonomy and the diversity came out quite strong. And then the last question, colleagues, was determine aesthetic characteristics such as child's skin tone or eye color. And again, although there was still a minority left, the vast majority against using the technology for this purpose. So I think our main observation is that context and culture are really important when deciding on the ethical and the legal aspects of heritable human genome editing. I look forward to your feedback. Thank you, colleagues.
Thank you very much for this very interesting survey. I had many questions indeed, but we're going to finish this particular panel before we get to the questions by turning our attention to Catherine Littler. Hello, Catherine, glad you could join us. You are our last speaker, and you are the co-unit head of the Health, Ethics, and Governance Unit at WHO in Geneva. You worked along with Alta as well on the creation of the WHO registry that I think we all would be interested in finding out what is going on with that. You focus on the intersection of ethics, governance, and policy in global health. And a lot of your recent focus has been on pandemic-related work. And you do indeed have the responsibility for a broad portfolio at the WHO, including realizing the potential benefits of emerging technologies in different settings. You were, as I mentioned, responsible for WHO's Committee on Human Genome Editing and are currently working on initiatives to implement the framework and accompanying recommendations. So I think that's something that I mentioned that I was particularly interested, but I'm sure we'll get back to that in the question and answer session. So the floor is yours.
Brilliant. Thank you very much, Jennifer. And I want to thank the organisers for the opportunity to speak. I'm going to just double checking everyone can see my slides. I'll take that as a yes if I hear no no's.
No, we still see the program. You have to launch or either duplicate screens in order that we see the whole screen.
You're seeing two screens, one screen.
No,
Catherine, just hit slideshow from first slide and it'll bring it into full screen mode.
Like that?
Yeah. Up top on your screen, you'll see the word slideshow in your menu on the top. Just under the orange bar, you'll see the word slideshow. Home, draw, design, transitions, animation, slideshow.
I don't have that bar. That's strange.
Yeah, what happens is that you must have a couple of screens. You have to duplicate the screens.
Okay, I've got now. How about now? What do I need? I'm on this
It's not ideal, but you can carry on like this.
No, hit slideshow and now from beginning, yeah. And it should bring it up into full mode. There you go.
That working? Brilliant. Okay, apologies all for my technical incompetency. It's lucky I'm not a scientist in this field, I think. So again, thank you to the organisers. I am gonna talk about the registry, but I really think I've just come off the back of the Paris Peace Forum, and we had a session there, and I'd really like to pick up on some of the key issues that actually segue nicely into what Doneric was saying with his engagement, contextual engagement. So I'm going to remind people of the work of the WHO committee, hoping that Alta was speaking about something else, as I'm sure she was. I'm going to focus on the challenges and the opportunities that we've seen with that governance framework and the implementation issues to date with implementing that and the recommendations. And then as I said, I've just returned from the Paris Peace Forum, and I really want to pick up on some of the key discussion points there, because for me, it was a really interesting experience, because often I give these talks to people who have an interest in this area or knowledge. And so it was really good to give talks to both potential participants in future trials, but politicians who aren't familiar too, so to get a really good idea of what are some of the questions that are coming from outside this audience in terms of the questions. And as Jennifer said, I will pick up on some of the recommendations and the registry and some of the challenges. But I'm going to move on. So some of this will be familiar to you all, but just as a recap, that the WHO committee was formed in December 2018. And in some senses, you'll remember the timing on the back of the summer of November 2018. What's interesting, actually, is we were already at WHO in the newly created science division talking about emerging technologies and governance and the ethics in corporation, but we obviously leapfrogged our work given what happened in the announcement of the November meeting. But I think that's important contextually to put that in there, because we started reactively, and ideally going forward in emerging technology, we don't want to be reactive, we want to be future-proofing, and I think that's a question for us all going forward. The focus is also on governance from the end-to-end processes, so very much from basic research all the way through to seeing through to therapies. And I'm going to argue again that that's critically important. We want to address issues such as access and equity. We can't just be thinking about access and equity at the time that we get to the point where we are accelerating or putting something into therapeutic. We have to be thinking about it earlier. The committee also, as you'll remember, came on the back of what happened in November. I mean, the original focus wasn't on somatic, but a lot of the conversations of the committee focused on somatic for a couple of reasons. One, because not only did the committee, but WHO perceived that the greatest health benefits in this area would come through somatic. So the greatest potential health benefits would come through somatic. And that was really important. And that in itself raises huge ethical issues if those potential health benefits are only available to certain populations. So that's an ethical issue in its own right. So that's really where the focus came from. And they built on existing initiatives and situated it in the broader emerging tech landscape. And that will be important because I'm going to come back to the relationship between human genome editing and other areas of emerging tech. But the committee also had a very broad remit, in the sense that not only did they have to understand the scientific, ethical, and societal issues attached to governance, but they were doing that at multiple levels, from the global to the regional, all the way through to the institution. So moving on, just to remind you again of the work, six committee meetings involving multiple participants, I think from 29 distinct groups, 4 working groups on particular areas, one of them being registry, one of them being the 3 E's, engagement, education, engagement, and empowerment, 15 online webinars with relevant experts from 71 distinct groups, all the way through from people who were experts on whistleblowing mechanisms and what happens to whistleblowers, because obviously that was one of the areas that the committee was interested in, all the way through to IP. We also had satellite meetings where we involved Indigenous populations, people from different disease groups. And I was really interested to see what Donneric's findings, because one of the participant groups we had was from the deaf community, and they really were against this because they didn't think that this needed to be done. So different perspectives from different potential participant or patient groups, but a very broad consultation. I think the one thing I'll come back to in a minute, and importantly, 2 online consultations on the governance framework, one early on in the development of the governance framework and one much later on in the governance framework development. And out of those came a clinical trials registry, which is attached to the CTRP, the International Clinical Trials Registry platform, a statement by the DG, and the 2 main recommendations. So one is the governance framework, which I've already alluded to, but the committee came up with 9 recommendations for governance and oversight, because when situating this work, the committee was very cognizant that any work on governance for human genome editing will fail if you do not start to address some of the systemic issues that are attached to it. And they're everything from unfair research collaborations to medical tourism to the IP issues. So I think the committee really thought it was important to draw attention to those, as well as the governance framework. I'm not going to go into this in detail in the interest of time, but the framework is built on ethical principles. I would argue that it's the first ethical governance framework of emerging technology in health in its space. WHO now have 2 other frameworks that have some similarities. One is the one on AI, and I'll come back to that because this is where we definitely see the convergence of emerging technology. And the other one is on the responsible use of life sciences, which took this framework as its starting point. So it really was a prototype and the first of its kind at WHO, and something that we want to review in due course. And I think the other key thing is, whilst it was the recommendations were for the DG, and obviously WHO is a member state organization, so we did want to focus on helping our member states with regulation and oversight in this. It really thinks of governance in the true collective response. It's not just about the responsibility of nation states, very much thinking about governance as an ecosystem. Again, just going through the ethical values, I'm not going to go through them, but what I think is carefully underpinned in the governance framework is all the questions, all the areas link back to the principles. So, for example, the registry, it's there because it's there for accountability, transparency, and inclusiveness. So everything links back to these foundational values and principles. I said that the committee really thought that that the governance framework would not work, and actually some of the key ethical issues in this space will not succeed. We will not address issues of equity and access unless we address some of these other recommendations. So they produced 9 recommendations, 4 of which I think will come up in the rest of my discussions. Obviously, I've already mentioned the registries, international research and medical travel, illegal, unregistered, unethical, unsafe research, intellectual property. This is a huge one, and I think we can learn a lot from the discussions that came out of COVID. And actually, some of the delays with this work is due to COVID have maybe benefited the work and benefited the discussions going on. And the big one that Doneric really focused on is education, engagement, and empowerment. And I'm really glad to see that a lot of this work is starting to be taken forward in different contexts. So what are some of the governance challenges that were facing the committee when they came along, and also are still facing us today? So clearly the fast pace of advancements, regulatory mechanisms, even in some of the best regulated settings in the world, cannot keep pace, right? So we need to strike a balance between legislation, regulation, to promote public trust and accountability in these types of technologies. and a bottom-up community-driven approach. And who is that community and how do we drive that? So who needs to be involved in the governance and how? And if this is a democratization of governance as well as research, how do you include all these parties? These are big questions that I think we haven't got the answer to. We've, in essence, with this governance framework, somehow opened those questions up and put them out for debate. A real democratization, as I said, of research and technology. And where that is challenging, I think, particularly for human genome editing, for the use of the tools, not the therapies at this point. I think we have to differentiate between the use of the tools and the therapies, because the tools are cheap and easy to use. So we've got people, the do-it-yourself community or the biohackers, you've got people who may not necessarily sit within the traditional governance institutes. They may not work in a research institute. They may not have funding from a traditional funder. So some of those mechanisms we can't use. So how do we speak to those people? As I said, the tools have been democratized, but not actually the access to the therapies at this point in time. And again, greater calls for inclusion in the development of the governance mechanisms. And we have to be honest as a community and say that this is a work in progress. We're getting better at it, but we still don't know what it looks like. I'm not going to talk about harmonization being the answer, but I do think this is where discussions, particularly across regions, are critically important. And we have some great examples from the African Union in other areas of technology where they've set up committees, for example, in gene drives, where they've set up committees to look at this from a regional perspective. So I think getting consensus at different levels is really important and helps with that, one, access and equity questions, and also helps with issues of stopping medical tourism or research tourism. So I think, and we cannot ignore the international nature of research. So we really, even though we have a lot of the power for legislation at the nation state level, I think we need to look at other mechanisms. Continuing the governance challenge, I think the engagement is really critical. In a lot of spaces, it's still a buzzword. I think that is changing. But what do inclusion of different voices in different settings look like? I think there is a tension, and we got some criticism when doing this work for including the word education as a bit condescending, but I don't think that. I think actually education is important. It's not just of populations. I would argue education of politicians. Interesting, at the Paris Peace Summit, I was speaking to some politicians that had little or low awareness of these types of technologies and some of the issues. And so I think the education is really important at very many different levels of society, and that does, over a period of time, lead to engagement and empowerment. But I think it is also critically important for bigger issues of trust in science, and I'll come back to that. I don't know what Alta or Donrick said about this because I missed most of their talks, but this discussion on what broad societal consensus means in a very polarizing world, I think it's really important for us to unpick this a bit more and think what consensus does need to look like in different settings, and how that consensus should fit into political decision-making when you have different types of political regimes. It's going to look very different in different settings, and what is our level as a community of acceptability. And again, we're operating in a space where no single body has the mandate. WHO has a lot of soft power in this space, but we don't have a single mandate, and nor do nation states. So really, when you have no single player with a mandate, you have a lot of players with legitimacy and responsibility. How do we make that ecosystem governance effective in this space?
Excuse me, Catherine. I'm sorry to interrupt you. I just wanted to let you know that you have 5 minutes left so that we can have a discussion. I'm sorry to...
No problem, I'll keep going. No problem. So implementation, I think, is critical. And since the last time I spoke to many of you, I think, which was at the 3rd Human Genome Editing Summit, we've had the implementation piloting 2-day meeting in Uganda on the back of the Ugandan country office actually contacting us that they're interested in doing sickle cell research in the country. They were worried about some pre-existing governance regulatory framework going through that wasn't about human genome editing, but had the potential of capturing this. And they were really worried that they didn't want to have this. So we had the 2-day workshop that we organised together with the country office, the Ugandan National Council of Science and Technology. We had 116 participants across a broad range, including patient groups, civil society representatives, research ethics committee, biosafety specialists, many different people from the bits of governmental departments. We looked at implementing the framework and developing a draft of the National Action Plan. We used the sickle cell scenario and associated questions, and it was good for framing the discussion for 2 days. The conclusions reached were a set of recommendations for governing in the Ugandan context. So I think by the end of the 2 days, we had a very good understanding of where the gaps, but where also the strengths in the Ugandan research regulatory environment were. There's a real need, I think, for continued public engagement and international cooperation to ensure safe and responsible use. One of the gaps, I think, that came out of the governance mechanism is we didn't focus on the safety and efficacy issues. And maybe going forward, that could be an annex, or if we did this again as another implementation case study, we would want to include a session on levels of safety and efficacy, and that came up again in terms of acceptability. So where does that threshold sit? Implementation next steps. Interestingly, we're collaborating with the South African MRC to host a meeting in February 2024. We are scoping additional country level case studies in the Paho and Sierra regions. And again, I want to emphasize the need for thinking about this on a regional level. And I say again, the African Union example. But the next development is we think it's really important to look at a technology agnostic, and I'm calling it a framework, but I'm not sure if it is an Uber framework or whether it's a document for us to work on. But innovative anticipatory governance of emerging health technologies. They are converging. AI is converging with, it will converge with human genome editing. So we need to really take stock of our current frameworks, because there's a real danger of contradictory governance to slow it down even more. So we are currently reviewing our current frameworks in this space, and we have a view to produce an overarching document. And from there, we will aim to test this in other fields, including neurotechnology, gene drives, and reproductive and developmental frontiers are the 3 areas we are proposing at the moment. That work will not start until 2024. And then finally, just some comments on the Paris Peace Forum. So for the first time, human genome editing was selected as part of the global health platform. So we had WHO had a stand there, so did UNESCO looking at their ethics work. The IBIS initiative had one, as well as the Global Observatory. So we were able to have people, as I said, who are unfamiliar with the subject area come up to us. We also had a panel discussion there, moderated by Hervé Schneinweis, and you can see the names of the people on that. Critical discussion points, I think, that came out. We had an Indian politician who was really interested in this, but he said he had more questions than answers. Really the difference between the role of societal views versus the medical. So what questions need to be asked, by whom and who is framing the questions. And this is a lot what Ben from the Global Observatory wants us to take stock of. How do we make allowance for context and different societal views? How to bring other voices into the discussion. And I would argue that the national ethics committees have a key role in promoting and understanding engagement in different settings. And actually this topic, well, the topic of engagement and trust in research, will be on the agenda for the next, so for the 14th Summit of National Ethics Committees next year in April. This is in their purview, and I think it's really important that they take case studies like human genome editing and other areas of emerging tech. We need to raise the level of this to a political dialogue. We're never going to solve some of the issues around access and equity unless we look at the bigger picture. There's a lot we can learn from COVID. We may have gone backwards in terms of global health justice, but it put issues of IP, access, equity on the agenda. We need to look at these, implementing regional power, development of infrastructure, and look at those as examples of areas that we could do here. Look at vaccine manufacturing hubs being built in Senegal and South Africa. Can we look at those models? of infrastructure, regional infrastructure in human genome editing, particularly in relation to therapy development. Registries, again, where are we with that? We have 144 clinical trials on the registry, mainly all to do with, they all start with the word safety, interestingly enough, and have CRISPR in them, the majority. I would argue if we're going to be good with the registries, we need them for transparency and inclusiveness. We need more investment in them. We need to moderate them. We need to check them. We need to update the terms, but they really do need investment. And so that's, they're only focused at the moment on clinical trials. There's an argument that you could focus them on basic research, but the ICTRP isn't set up for this. And this is my final slide, so I'm almost there. We do need a cultural shift in research practice and our approach to governance. I think that's starting to happen. That happened at the 3rd Human Genome Editing Summit in March. We had a whole day on issues of access and equity. That was not at previous ones, but I think we need to draw more attention to this. And we do need to situate the work we're doing in the broader emerging tech landscape and look at better tools for monitoring. And I will stop there. Thank you very much.
Thank you so much, Catherine. And again, my apologies for interrupting you. So we have about 20 minutes now for a discussion. And I am going to go straight to the chat box and then just save my questions for later. I have a question for Dr. Thalder. Thanks for your very interesting survey. How do you think this could influence health policies in South Africa? How are the results, or were they communicated?
Thank you for the question. I will put the link to the article in the chat or in the answers. It was both the study protocol and the study results were published as articles in PLOS ONE. And regarding how it affected policy development, well, that must still be seen. Of course, my research group is quite active in this space, and we have integrated the results of this public engagement, of this deliberative public engagement, with our other conceptual work. And we have made, we've already made suggestions to government on how to implement it in actual policy. But you know, the wheels of government turn very slowly, so we will see. But I'll share a link to the article to the article in the in the Q&A.
Okay, a question for Catherine Littler from David Curry. Very enthusiastically asks you, in terms of the next near-term normative milestones and real action in governance of human genome editing, somatic, heritable, shouldn't or doesn't this rest with the WHO? And in essence, he argues, what is the next step?
So it's a great question. Thanks for that question. And you mentioned in there the WHO, and I think this is a competition between I myself, as a technical person at WHO, cannot raise anything that goes into the WHO. That has to be raised by member states. So, David, I would argue if you think this is something that WHO has to take on at that level, then that has to come through the member states. I do think that this is a combination between us working at headquarters to promote this work with our regional and our country level counterparts. And I think the Uganda worked really well because the need came from the Uganda country office. So we were addressing an on-the-ground need. But I think it sits between the three regions. But as I said, if you do think this is important and we do need a mandate, and once you want this to go through the World Health Assembly, then that does come from the member states. And I would argue that if you work with your community and push that through the member states, we would welcome it.
I see, very well. A question for Dr. Thalder by Didier Cornel. Very interesting, thanks. Concerning enhancement, didn't you think about asking about longevity beyond what is now possible for humans? Did you think about putting that in your I was
actually just now typing an answer to
okay.
the gentleman, so I'll quickly answer it verbally. Excellent idea. our problem was, of course, just that we had limited time. So our group then decided what are the typical issues that we saw most in the literature. And we had to limit it to 5 questions regarding enhancement. And as you would have seen, we've also tried to combine some of them, like intelligence and athleticism, which is also debatable whether that was really a good idea. But I think this was just meant to get a general idea of how the South African public feels about these kinds of enhancements. I think if one is really interested in enhancement, then one can easily have a whole event just on that, because that is actually so complex and with so many different possibilities and different potential ethical positions on the different kinds of enhancement. But that wasn't our focus really with this study. But thank you very much for that comment.
Okay, I have a question for Alta, and she also wants to say something. So I'll ask my question, and then she'll answer my question, and then she'll say something, and then I'll turn it over to Luis. Alta, my question has to do with the difference between public-private financing of human germline editing. Now, you said, rightly so, that there isn't a law against it in the US, but there's this sort of trap that researchers cannot get out of because of the way the FDA sort of oversees this. What about private companies? What do? They don't need any type of approval from the FDA to do research up until, like, say, 14 days, right, on embryos.
Oh, no, yeah, research, preclinical research.
That's right.
Is not a problem. And in the US, the only issues on preclinical research have to do with any funding constraints. And there are such when it comes to federal funding if you're working with human embryos, but otherwise, no. But the point is that private companies, just like publicly funded researchers, are all subject to the Food and Drug Administration's authority. So yes. A clinical trial cannot begin without FDA approval. So research, before you try to actually transfer an embryo for gestation, okay. But the moment of transfer is the trigger for the FDA's claim of jurisdiction. The question I was going to ask is actually about the South Africa survey, and specifically on some of the enhancement options that you offered people. It seemed to me that saying it's conditioned on safety and efficacy supposes that there really is the ability to genetically enhance some of these traits, but intelligence, aggressiveness, some of these things that you offered people as a thought experiment seem to be very far from reality. And I wondered how you decided to offer those as a discussion point.
Yes, I think we made it quite clear to people that although these applications, if I can call it that, are indeed discussed in the literature, it's not at all certain that that it will be ever possible for our genomic scientists to influence intelligence. But what we do know, of course, is that there is a genetic basis for intelligence, and most probably also for something like sexual orientation, and most certainly for things such as skin color and eye color. So we explained it in the thought experiment to them that this is not to say that it can be done or that it will be done, but assuming that it can be influenced, that the technology can to an extent influence it. And then, of course, the normal proviso, namely that it must be safe and effective.
Luis, and then Francois.
It was not assuming.
Sorry.
Yes.
Yeah, I just see the facial reaction from Alta, and I don't know whether my answer was to her satisfaction. And,
it is. I mean, I appreciate it. I worry that we're giving people, no matter what you tell them, the impression these things are doable and that they are strongly determinative. And I worry about creating that impression, since it then gets people debating these topics based on a misunderstanding. But I appreciate your effort to give them the nuance.
You can be assured that there was no misunderstanding, and that the material that they were provided with were not only drafted very carefully by our research group, but that it was also peer-reviewed by international people. So I'm very certain that there was no misunderstanding at all.
Okay, thank you. I'm sorry to have interrupted. Luis.
Oh, thank you, Dorrick. I will be elaborating further what Alta was posing, because I was a bit concerned when I was listening to some of these topics, and beyond what has been already discussed, we certainly, and we have to say this very clear, we don't have the genetic basis of aggressivity or intelligence. We don't have this. But what we also don't have is the genetic ability to interfere or interact or to correct Down syndrome. So Down syndrome certainly is something that we cannot attack at the moment. It might be doable in the future, but at the moment, so this is a large chromosomal anomaly, which can be variable. And this is, so asking this to people, it provides the idea that this is something doable, and this is I'm a bit concerned. But beyond this, you were giving an example. I happen to be working on albinism since many years. And albinism is kind of one of the cases that Alta was referring to, because albinism is characterized by this hypopigmentation, which is treatable. So you can protect yourself from sun, and you can treat with cream, and you can treat the skin lesions. But what is not treatable so far is the visual inability, the visual handicapness. And there we know that the window of time that you can do something, the therapeutical window, it's about up to 2 years. So essentially what you would like to do is fetus therapy. So when someone is going to be born with albinism, you would like to be able to deliver these genome editing tools during the fetal development. And I wonder whether all these details were probably not provided to the people. By the way, pigmentation, even though it looks apparently easier, there are as many as 700 genes affecting pigmentation. So it's not as easy as activating or inactivating 1 or 2 genes. Thank you.
So nobody is assuming that it's easy, and nobody said it's already safe and effective. It was made very clear that we're not trying to create policy for a technology that's being applied now, but that we do think that it is wise to start to involve the public and gauge the public opinion, not stakeholder opinion, because that's also something different, but to engage with the public opinion on these issues well in advance, whether it might take 10 years, 20 years, or 30 years or more. So that was our position, to look at a time horizon in future and then say hypothetically.
And I appreciate, if I can say, I appreciate the fact that you wanted to share that there might be different answers according to different environments and different cultures. And this is something which is absolutely valid. And I think this is something to take into account.
Bonsoir.
Yes. Thank you, Jennifer. It's a question for Katrina. Hello, Katrina. In the previous session, David Curry asked a question. I read you the question. A recent Lancet Commission hematology on sickle cell disease suggested the 2040 horizon before meaningful, affordable genome technology access. And I asked the 2 panelists from the previous sessions, one is the CEO of a company developing gene therapy approach, and the other is developing gene therapy in eye disease. I asked them whether they see a future to the transfer of technology in low and medium settings. So what's your feeling about that? Do you think it's feasible, and in which timing?
So if I understand you correctly, Francois, the question from David was that the timing now is that we won't get to an affordable level. Is this for people in low resource settings until 2040? And is that acceptable? And also, is there a market? Well, there's clearly a market. I think the question is, how do we incentivize, not criticize, how do we incentivize companies to go into that market and reduce those costs and bring them down faster? And this is not a unique question to human genome editing. We have this going on in malaria. We have this going on in different settings with vaccines, right? So rather than calling out, criticizing the people who have developed the vaccines, we want to encourage more people to go into the market. So I think there's a lot of work, and WHO, my regulatory colleagues, my other colleagues have a big role to play in this, but we need to make the ethical arguments. But we also have to be savvy about this. This is an economic question as well as an ethical one. This is around IP. This is around a whole bunch of others. We need to look at the political momentum being gained from COVID and translate that into the human genome editing field. Is it acceptable that it's not until 2040 we get that model down to affordability? No, even within economies of scale argument. But how do we incentivize? And I'm sorry I missed Alta's chat, but about the public-private sector, because it's going to have to be the public-private sector. And we're naive if we think that isn't the case. But how do we come up with better incentive mechanisms to get people in this space? Because there is a business model at the end of the day. What is the prevalence of sickle cell? It's high, beta thalassemia, these things. So we really got to think about it, but it's the cost at the beginning that it's hard. So I worry that some of the discussions we have at the moment really disincentivize people going into this space. We really want to think of new models and new ways of incentivizing. And I don't think this is an easy question. And I wish I had the brain capacity, and it's above my pay grade as well to do this. But I think we also have a role to play in it. This community can also think what we could be doing and the buttons we should be pushing. But that's why I enjoy going to the Paris Peace Forum, because I think that this is a political question that we have to get on the agenda. And we're a small community, but the impacts for public health could be great. So I don't have an easy answer, Francois, but I think we have an opportunity post-COVID to push this, and we need to push it.
Thank you.
Thank you, Catherine. Alta wanted to say something, and before you do, I have one last question for you. I don't understand this privacy argument that Dr. Hu puts forth. I mean, when you read a scientific article in a peer-reviewed journal, you don't have the names of the patients. Why doesn't he just, to erase all suspicion or what have you, why can't he just publish a scientific article about what he did? The privacy of the daughters of the girls will not be revealed.
Well, first, just to check, Jennifer, I thought I saw Francois Hirsch's hand up before mine. Did you?
I'm sorry, Francois.
No.
Oh, okay.
No.
With regard to your question, I certainly can't answer it, because the last time I spoke to Dr. Hu, it was the night before he gave his presentation. I have not spoken to him personally since then, and he's refused to answer questions. So one can only speculate.
Okay.
But you're right that you can certainly try to respond to requests for information without revealing identity. No question about that. On Catherine's point about the financial incentives, in addition to the experience with vaccine development and public-private partnerships during COVID-19, another area that might be worth looking at is the area of antibiotics development. Antibiotics have the interesting characteristic of being products that we desperately need to have, but not to use. We need to have antibiotics that are not overused, so that in cases of resistance, you have something new to try for a patient. And so the usual business models don't work, because you want companies to make antibiotics, but then not really sell them. And so it's another example of an area where you have fundamentally a market failure in the current business model for the kind of development of new therapeutics. And the Infectious Disease Society of America has been talking about this for decades. And we're just beginning to see a few new proposals, at least in the US Congress, for providing pre-approval incentives to de-risk the development process and make sure that companies aren't at risk of losing tremendous amounts of money on the way to developing things, even before they find something that works that might be the subject of let's say, a government stockpile purchase. But it just seems to me that for people that are more economically savvy than I am, certainly, it's not my field, we need to be looking across fields at areas of market failure and asking what are the commonalities here that we might use to inspire us to come up with a new solution for the area of gene therapy.
Thank you, Alta. Luis, do I turn it over to you?
Yes, this is 1 minute left for discussion. I wanted just a comment and a quick question to Alta. So definitely, I'm afraid I think we will be having another several Dr. Hers that will be appearing, maybe in other countries. And the question that is left is, how are we going to be handling this? What are we going to do, the rest of the world? Because we cannot block these things. We cannot prevent this from happening. We can say whatever we want, but this will be happening anyway. And so is there anything left that we can do besides isolating these people and stating clearly that this shouldn't happen? Alta?
You know, Catherine mentioned the whistleblowing discussions of the WHO committee's work. One thing that we talked about that has not yet been implemented, I think COVID kind of got in the way of a lot of things, was to try and rally the resources of not only national regulatory agencies, but international and national professional societies, so that WHO might in the future play the role of a catcher to receive complaints about concern that so-and-so is looking like he might want to do something unethical, and have WHO figure out who might be able to have some authority, whether it's a professional society or it's an academy or it's a regulator, and send the information forward. WHO cannot be the disciplinary body, it cannot be the investigatory body, but it may be able to play the role of an intermediary to funnel information to those that might be able to have authority to try and discipline those who are going to violate, whether it's professional norms or actual regulatory rules.
Thank you, Alta. Maybe Catherine wants to say something briefly.
Just wanted to add something quickly, and I think this is where soft law and soft power actually can come in and make a difference. And that is when the committee was deliberating, somebody else came up and wanted to do a similar experiment, but in a different area, and the committee became aware of it, and they wrote to the DG and said that this shouldn't be done. The DG then wrote to the Ministry of Health in that country, and that research was not done, right? So I think there's a place for soft power as well. The other thing I want to add on to what Alta said is, I think we need to create trusted spaces where scientists actually talk about what they're doing more, because we know a lot of people knew what experiments were going on and no one felt they could say anything for cultural reasons as well as other reasons. So I think we need to create spaces as well. This is a cultural shift we need to create. A whistleblowing mechanism by itself isn't enough. A change in the way we do research also needs to happen. So these kind of safe spaces, it's hard to make a nice recommendation about that, but I think over time we need to make that a big concerted effort to do that. Really out this sort of thing.
Thank you, Catherine. Thank you all. I think we have to move on and then try to finalize this. This is very interesting third Albrecht Müller Seminar. And we have asked our colleague Julian Kinderlehrer. And Julian was a former president of the European Group of Ethics, and he actually, at the moment, he's serving the ARRIGE board. And Julian, you have the floor, and you can sort of wrap up and present us with your summary of what you have been hearing. Thank you, Julian.
Thank you very much, Luis, for helping to organize with Sheetal and Francois a superb seminar. It's been fascinating. I've learned a lot and thought about a lot. I think we have to think very carefully as well as going back into history. Medicine doesn't stand still. New technologies have modified our ability to provide alleviation for many diseases very quickly over the last, let's say, 100 years. It's amazing what has been achieved if we think about the COVID vaccine being produced in hardly any time at all. That which we have now, compared to the technologies available before the Second World War, is simply amazing. And we have to recognize that. Is the introduction of new genetic technologies simply a continuation of this drive within medicine, of that which has developed over the last century, or is this really a massive step forward? I'm not sure, and I've been fascinated by the discussion today. Secondly, from what I've heard, all technologies involve risk to the patient. We can, to some extent, identify the risks and their probabilities, and we can explain that to patients. So, should the possibility of harm stop the introduction of a new technology? I don't think so, but we have to think about that very carefully and compare these new technologies and the risks of current technologies that are being used. If the current technology imposes relatively little risk, or if this new technology rather gives us relatively little risk, can we afford not to use it? Is it ethically acceptable not to use the technology? We talked about non-financial cost to the patient, not the financial cost, but the non-financial cost. We have to think about that. We heard the implications of stem cell replacement, for example, earlier, involving very long hospital stays and the issues of continuing immunosuppression for the patient. Is that acceptable? Do patients need to think about that as well as the academic who applies the technology? The financial cost to the patient was also raised. I think I heard a huge cost of many millions to the patient or to the health system, which might be asked to provide that medication for the patient. Can we afford it? If the cost to the patient for sickle-cell disease is so high, which we heard this afternoon, can we afford to use the technology for anything else? Certainly at the moment. What about the cost when things go wrong to the health service or to the health practitioner? It's a major issue which we have to think about. And finally, on that, all I've talked about now is therapy. It's not heritable therapy. If it is heritable, obviously, we have possible harm, not to the patient who can be asked a question, but to all their children and future generations. They cannot give permission. How do we handle that in our society? So there's so many questions which I found absolutely fascinating this afternoon from so many different angles. Thank you all for an amazing seminar.
Many thanks, Julian, for this brief but intense and concise summary of what we heard. Let me thank all the 6 panelists that have been with us this afternoon. And of course, Francois and Jennifer, who were chairing these 2 roundtables, and Francois and Sheetal for organizing, and everyone who attended for their interest. As I said, we will be editing this webinar, and very soon it will be published in the YouTube channel of ARRIGE. Thank you very much, everyone, and have a nice rest of the day. Bye-bye.